Well, good morning, everybody. It's good to see y'all. It's been a few weeks, so hopefully things are going awesome and lots of field visits are happening and soil sample installs and all the goodies. We'll give everybody a minute to get connected and get settled in. All right, great. Welcome. Good to see everybody. So, we're gonna just quickly do an announcement or two, and then we're gonna turn it over to our awesome partners at CSU to talk a little bit about soils today. So that's great. Just a quick reminder, make sure you fill up those soil sample bags, and Wilma's probably going to dive into this, but we're looking for about 3 pounds in there. Also, make sure you're putting the soil using the protocol into the little microbiome tube, and we're going to hear about that this morning as well. So, Miller, do you have anything you wanted to add here right quick?
Nope. I think we want more than 3 pounds, right? Fill those bags up.
Fill them up.
That's what we're after.
For a reason. We use those big bags, jam them full of good soil. So, and Wilma, you can kind of go through some of that in your presentation this morning about what you're looking for and what you're doing, probably. So, all right. So we have awesome team of folks here from CSU, including Wilma, Megan, Lexi. We also have Chris and Laura is here as well. So we're gonna turn it over to them and get them going on their presentation about the different metrics that are being measured and what's going on with that microbiome sample and what the report is actually going to look like so you can understand that. And just one last note is that we're not going to get these tomorrow or the next day, right? It takes time to get these processed. So that's why we're doing all our installs. We'll look forward to seeing these, and then we'll circle back with those producers and visit with them about some of these key things that we're going to discuss. So with that, I'll turn it over to y'all. Thank you so much for helping us out with this morning.
Okay, I'm gonna share my screen. Give me 1 second. And let's go into presenter. Can we all see that? Are you guys actually seeing the slideshow, or are you seeing my presenter view?
Looks good.
Yep, looks good.
Okay, awesome. So great. Glad to see you all today. Good crowd. Today we're going to be talking about soil health analysis and interpretation and what the CSU team is doing with all of these samples that you guys are going to be taking or you have been taking. So just quick to introduce our team and all the people who are going to be speaking today, because we've got a lot of people talking for our different expertises. I'm Dr. Lexi Firth. I'm a postdoc at CSU. I work on the soil health team in the soil health lab and am affiliated with Enriches, which is the Integrated Rocky Mountain Region Innovation Center for Healthy Soils. And I did have to write that down to remember it. So blame Megan and other people for naming that. And I'll let the rest of the team kind of jump in and introduce themselves.
We can blame Jim Ippolito for that because he's not here. But yeah, I'm Megan Mockmiller. I'm going to keep my video off. I'm sitting on a rangeland in South Park in my canvas tent, and I haven't showered in a few days. So I'm doing you all a favor just to give you my showered version picture there. I'm a research scientist at CSU and have been working with this awesome team. And we'll go into some of the soil health analyses that Wilma is leading at the Soil Water Plant Testing Lab. Wilma.
Yes, I'm Wilma Trujillo, and I'm the director of the Soil Water and Plant Testing Lab. That lab was moved from Fort Collins to the new facility at CSU Sport in Denver. And I'm gonna talk about the reports, what were you finding in those reports and how to interpret those reports.
Laura?
Hey, I'm Laura Mason. I'm a postdoc at CSU with Kelly Wrighton's group. I'm a microbiome scientist, and we'll be talking today about how we'll be incorporating microbiome data into future projects and models.
Chris?
Hey folks, I'm Chris Corpenning. I'm a master's student. I'm also a Dr. Kelly Wrighton. That's kind of our microbial ecosystems lab in the soil and crop science department at CSU. So yep, I'm a master's student right now. I received my undergraduate degree also in soil and crop science. So I'm sticking around. Glad to be here.
He's a rock star. He's leading a lot of stuff for us. We appreciate him. All right, well, just to get started, soil health. What is soil health? I feel like everyone here has seen this definition 1,000 times at this point, but Megan and I were talking about how we just have to start presentations with this because it gets our like for our own sake to get us into the mindset. So soil health is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans in a socially and economically productive and sustainable manner, which is a great definition, but it's not very specific, right? Evaluating soil health can be really difficult. There are practical challenges to consider. There are just number challenges to consider because soils are complex and not all soils are going to provide all of these ecosystem services equally or simultaneously. And it's not going to look the same from place to place. It's contextual, and they're not representing identical capacities to function across a landscape. So we start with that definition to say that this is an ecosystem we're trying to evaluate. It moves and it changes, and sometimes it's a little hard to capture. And we're going to do our best, but just like keeping that in the back of your minds that what you're seeing at one point in time may not be what you see 2 months from then, or like at a different point in the season, or after a different rain, or after you fertilize. But we are measuring quite a few things. Soil health is the biological, physical, and chemical components of soil. It's like that little center part. So we've got a big list going on here. The chemical is probably where most people are familiar with. Carbon, nitrogen, phosphorus. We're also measuring some physical attributes. That's available water holding capacity, texture, water stable aggregates, and then more biological components, which is going to be your PMN, and other nitrate and nitrite characteristics, because those change very, very quickly. So this is a huge, long list, and it is kind of important to point out, if this will change screens, that, whoops, my bad. This list of properties, like some of them are going to be inherent. They're going to be things that you cannot change. So when you're talking to a producer about this, you're not going to be able to change your soil texture. That's what you are. That's where you're at. You're not going to be able to change your climate. That's just what you were given. You're not probably going to be able to change how much water you're allowed to have on your land. And then there's going to be the more dynamic properties that you can change over short periods of time or with management. And that's going to be things like the structure, the porosity, like how much water can you hold? Like how do those aggregates form together? It's going to be biology, which is going to change with management, change with the time of year. And then when you go through the soil test, there are going to be some things that's a little bit of a gray area, this like sort of inherent, sort of dynamic metrics, because like, for example, cation exchange capacity, right? Clay tends to have a very high CEC because there are a lot of negatively charged sites on clay surfaces. So inherently, clay tends to have high CEC. Sand, if you have very sandy soil, it's going to be low. But if you can add more organic matter to your soil, which has a lot of negatively charged ions, you can shift that cation exchange capacity. And so understanding how all these things are like fitting together, and they can change and shift through time, and being able to provide that context for a producer is really important. And I'm actually going to hand this off to Wilma now. She's got her own slides because she's the one that's actually running all the tests and is going to be delivering the test reports that you're going to get back. So I will stop sharing. And Wilma, if you want to jump on.
Hey,
Thank-
Wilma, I think you're
Yeah.
muted.
Okay, sorry
We can
about.
hear you now.
Okay, so I'm gonna start talking about sampling before we go into the report, what the report will look like. And I want you to remember one thing, that the results of a soil test will be as good as that sample had been taken. So when we talk about give you a protocol for you to follow to collect that sample, that was for a reason. And so we want you to collect the sample year after year the same way. So that means you're gonna go to the same places where you start in year one. You're gonna go and collect sample from the same depth, and that depth is 0 to 6. It's not between 0 and 6. It's the whole column of 0 to 6. And as I say, go back to the same places. Today we have phones on our hands with very good GPS, so we can mark in our phone that site where you collect that sample. Also, we want you to take more than one sample in an entire field because there is a lot of variability. So we want the sample that you send to the lab to represent that field. And the only way to do that is that you collect multiple samples from that field. Once you collect that sample, you're gonna mix it well, and then you take that 3 to 5 pounds or whatever it feed that bag that you guys receive. Less than 3 pounds is difficult to do all the tests that we are doing. You saw that long list of the tests that we're doing. So that's the reason why we need the muscle sample. So let me go now and share my screen to talk about what the soil test is gonna look like. So the sample ID is a number that we assign to that sample when it comes to us. This as an internal ID for us. Then we have a sample ID. That sample ID is I'm following the producer's number, the name, and the conservation district that they come from. Soil depth is that 6 inches because we need that depth to be able to calculate the amount of nutrients that are in pounds per acre that are in there. So let's talk about the first, what is soil pH? Soil pH it only tells us how basic or how acid a soil is. And we have some ranges here. And this bar of color is only indicated where your result, that 7.8, fall in that scale that we are giving. Okay. Soluble salts or electric conductivity. This is not related to the sodium chloride that everybody knows when we talk about salts. This is organic and inorganic salts in the soil. means compounds that are in there that are soluble, right? So a plant, some plants are more tolerant than others to salts. We like to see you guys below this 2.0. We like to be there because if there's higher soluble salts, the plants, even so there's sufficient water in the soil, the plants are not gonna be taking water. They are gonna be competing, the salts and the water, and the plants are gonna be competing for that water. So the plants are gonna be suffering from a physiological drought because it's too salty. The plants don't like to take salty soil and salty water. Organic matter is everything that is organic in that soil. Most of soils in Colorado are about 3% or less. Another thing that I wanted you to know is this column here, this row here that say 1-to-1 or loss in ignition or 2M potassium chloride. That is the methodology that we use to process that sample. For pH is 1-to-1. For soluble salt is also 1-to-1. That means it's one part of the soil versus one part of water. Organic matter, we do that by loss in ignition, is putting this sample at 350 degrees for 2 hours. And that is a measure of organic matter. That includes everything that is organic. It's not just carbon, everything. Nitrate. Is the nitrogen is that the most important nutrient for plants. Plants love nitrogen and are gonna take a lot of that. And here we have some different range, right? Very low to very high. And also we're gonna take your result that for and put it, rank it and see where we are, right? Notice that there is not an optimum because for some plants take more than others. So that for that reason, there is not an optimum there. Ammonium nitrate, ammonium nitrate, that's another form. Those are the mineral forms of nitrogen. Those are the only molecules of nitrogen that the plant take, either nitrate or ammonium nitrate. Then we have phosphorus. Phosphorus also, there is an optimum because phosphorus, this measurement is an index. So plants need about 11 to 15 parts per million of phosphorus to meet their requirements. Potassium, we like to see plants about more than 160 parts per million. Calcium, magnesium, sodium, those are the nutrients or the minerals that we need to calculate that cation exchange capacity. What is cation exchange capacity? Cation exchange capacity is that capacity of the soil to give to the soil medium or to the soil solution nutrients. Nutrients are attached to the minerals in the soil. When I say minerals, I mean sand, clay, silt, right? So the nutrients are attached, bind chemically to the minerals, and the capacity of the soil to give up to the soil solution is to maintain a balance. That there's always a balance in the soil solution. When the plants take and the soil go back and put them back into the solution, right? So there's always a balance. And the higher the CHC, the best is for the plants. So that means that when the plants take, the soil is gonna release more into solution for the plants later taken. If you have any questions, don't doubt to stop being an ask, right? So now talk about micronutrients. Micronutrients are needed by the plants in really small amount, but they are essentials for plant growth and development. So it's not like nitrogen, phosphorus, and potassium and sulfur. They are all essential. There are 17 nutrients that are essential for plant growth and development. But what we divide them in macronutrients and micronutrients. So those macronutrients are those that the plants are gonna take in a huge amount. And the micronutrients are also essential, but the plants need little amount. These zinc and iron are pH dependent. That means that if we are in alkaline soil, some of these nutrients are gonna be less available for the plants. They are there, but they are not plant available. So it's important to know, because another thing is the yield potential. If one of these nutrients, nitrogen, phosphorus, potassium, sulfur, zinc, and iron, are deficient, if one of these nutrients, just one, is deficient, plants are gonna be having a problem to grow and develop. Only one nutrient. So what we trying to do this here with a soil test is to see what of those nutrients are in a level that the plant is not deficient. Then we have, in this part of the report, we're gonna have the nitrate and ammonium that are mineralized after 28 days in incubation. So this mineralized nitrogen, ammonium, and nitrate will tell us a little bit of how active the microorganisms are. If we get a significant amount of nitrate or ammonium during this incubation, we know that the microorganisms are doing what they are supposed to do, mineralize or decomposing that organic matter and putting some nutrients in the forms that the plant can take it. Another measurements that we're doing is available water holding capacity. This water is the plant available water, means is the amount of water that is available for the plant to take it. There is two different forms of water in a soil. There is a field capacity water. That field capacity water is that is not tied up, and it can be lost through evaporation, through evaporation or gravity, right? And there is another measurement that is the wilting point. That wilting point, that water that is a wilting point, is the water that the plant can take it. So the difference between field capacity and the wilting point is the water that it's available for the plant, that water that the plant can take it. Soil texture will give you a percent of sand, percent of silt, and percent of clay. And also we give a name, soil class, like in this case, a sandy clay loam. We also gonna look at total carbon, total organic carbon, total inorganic carbon, total nitrogen, and a carbon-nitrogen ratio. That carbon-nitrogen ratio, what it means, that carbon and nitrogen ratio. That means is the relationship between the total carbon and the total nitrogen. In soils, that is more or less like between 10% and 12%. In this case, we have a 12%. Water stable aggregates, what that means, what is a stable aggregate? Is that aggregate, well, there is a misspelling there. An aggregate that will support, how do you say, withstand water erosion. If the soil is well aggregate erosion, you're not gonna have a runoff with sediments going into the ditches. If the water is well aggregate, that soil is gonna keep it inside. It's not gonna run off. And the sodium adsorption ratio, that's another spelling there, is the relationship between sodium, calcium, and magnesium. And that is gonna tell us, the higher this number, the worse it can be for soil structure. If there is a lot of sodium in the soil structure is gonna be damaged. If the soil does not have that structure, if there's no those aggregates, soil is gonna be compacted. Any question? I think that's all I have. If anybody have any other question, please just email me about the report.
Yeah, well, I do have a question for you on that SAR. You know, a lot of times there's some different targets. I know it's based on soil type and other things. Do you have some suggestions on what some upper end limits are on if you see sodium absorption ratios that are above a certain level? It's something that we need to, you know, start thinking about how do we address that?
Yes. If it's more than 13, you have a problem with sodium. And that's when it needs to be reclaimed. And we need to do another test to calculate the amount of gypsum that we need to apply to reclaim that soil. In Colorado, gypsum is calcium sulfate, right? We have, I think we don't have a line estimate in the test, but in Colorado, we have, in addition to alkaline soils, we also have calcareous soils. There is a lot of calcium. And so because of that amount of calcium that we have, free calcium, that's a line, right? Free calcium, reclaiming those soils is very, very, very difficult. One thing that I want to make a difference is between electric conductivity. We have saline soils in Colorado, right? We don't have that many sodic soils. And sodic soil is when we have a SAR that is greater than 15 or 13. What we have problems in Colorado is here. EC, electric conductivity. This is all salts.
Would you be able to describe a little bit more about the difference between the salinity and sodic? Because I think some of us might like to know how that differs, if you don't mind.
Okay,
Thank
EC,
you.
electric conductivity of soluble salts. This is all salts, organic and inorganic salts. When you fertilize, that's a salt, how you fertilize it, right? So we have, you apply urea, right? Or ammonium. If you apply more than what you need, you will have high EC, right? And the only way to get rid of high electric conductivity or high soluble salt is to dilute them. That means applying excess of water. Okay? Now, what is SAR? SAR is that relationship between calcium, magnesium, and sodium. I'm not talking about any other salt. Do you see what's the difference? This is total salts. EC is total salt, organic, inorganic salts. SAR is only calcium, magnesium, sodium. In this EC, you will have nitrate, phosphorus, sulfur, all those salts. This is only the relationship between calcium, magnesium, and sodium.
Thank you for doing that, Wilma. Looks like there's another question about when would soluble salts be too low? Are there instances where that might be too low?
No, it's better to be very low than being moderate or moderate high, right? Because if you are higher, you don't want to have salts because plants don't take salts. Plants take anions and cations, right? So what plants take nitrate, plant takes ammonium, phosphate, potassium plus, calcium plus 2, magnesium plus 2. Sodium is not an essential nutrient. Zinc is plus 2. Iron could be plus 2, plus 3, right? Plants don't take, let me think about ammonium sulfate. That's a salt. Ammonium sulfate is a salt, right? For the plant, the ammonium needs to go alone and the sulfate needs to go alone. And that's what the plant is gonna take. Is that clear, the difference?
I think so. Penny, please speak up if it's not clear to you.
I think it's clear enough. When we go over these or get these soil sample results, are we expected to go over these with the producers, or who will be the one delivering the results and explaining this information to them?
I
Yeah,
will
I
be
can speak.
available to explain the results to producers and farmers, farmers and ranchers. But I don't know what the CDA have in mind.
Yeah, well, I'm happy to speak on that. And Penny, this is, there's going to be a lot of these coming out, and we're going to be doing those one-on-one visits with every producer. And so we're happy to help have those discussions. There's a lot of things that we can point to, and a lot of things that we are, and you guys are going to hear a little more about things that we don't know a lot, and we're trying to learn more about. But it's real specific based on what their soil texture is, what their crop rotations are, and all those things. And so our staff is available to help you interpret that, and we're more than happy to help talk through some of that stuff with those producers who have those questions. So it's going to kind of be a team effort, and we'll all kind of share the responsibility to help producers navigate these useful results. And just one last thing to remember, this is our baseline for improving the soil health, right? So we're starting here. We're going to use these for comparative analyses, but we're also going to be able to do some different discussions about what they're doing and how they're doing things and how we can help them maybe do a little better based on what they're trying to grow.
Thank you.
So I have a question on the organic matter. You said the Colorado soil average was 3. Is there any way to get that higher?
That's what the soil health improvement is all about, trying to increase organic matter, right? But organic matter is difficult to increase it. It may take a whole decade to increase. And the limitations that we have in Colorado is water. That soil organic carbon, that carbon comes from photosynthesis, right? And so we need to increase production. We need to increase the amount of crop residues that stay in the soil to be able to increase that organic matter.
Yeah, Wilma's right. And, additionally, we know that certain soils can only kind of handle so much percent organic matter, right? There's a limiting factor in those for a lot of reasons. But we do know that some soils in Colorado, go to an aspen stand and take a sample underneath those. They have extremely high amounts of organic matter. Now, but it does take a lot of time and effort. And I have seen some that are, 8% on ag soils. So we can get up there. But it is a really hard thing. And we know that just incorporating compost isn't the #1 way to do that. Like Wilma mentioned, the stubble, there's also, the plant roots, the residue, all the other things that are part of our soil health program will help us increase that. But a lot of it relates to, making sure we have that good structured soil so that our biological community can process that organic matter and turn it into something that's, stored in the soil. So,
and
we all dirty discussion there.
We also know pretty clearly that tillage and disturbance burns up organic matter. I mean, it's the #1 enemy, right?
Great point.
Yes, reducing tillage will at least maintain the organic matter in the soil. But one thing that I need you to understand the difference between organic matter and total carbon, right? That's what I say, organic matter is more than carbon, right? Organic matter is everything that is in a plant residue, right? So you look in this example that I have a 2.2% organic matter, right? But I go back down here in total carbon, this 0.781, that's total carbon. That's the carbon that we need to increase. That's the carbon that needs to be sequestered and protected. But how we get this? From here, from soil organic matter. That's our soil. And that is photosynthesis. That is producing a crop. That is leaving that soil crop residue on the ground. So I'm gonna stop sharing so the rest of the presentation can go on.
Great. Thanks, Wilma. That was awesome. Sharing screen again. So Wilma went over most of this, but I just want to reiterate a few things. We do need 3 pounds of soil or more. We're running low on some of these samples, and it means we can't analyze everything that's on that list. So please, 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 minimum 3 pounds. Remember, please take your sample at the whole 6-inch depth. So you have the whole spectrum, not just from the top, not just from the bottom. And also take the GPS point of that field. And also we are to that survey 123, or if a survey you all have been filling out with every sample you take, we are going to add one more question to it that I just want you all to be aware of. We're asking if that field has been fertilized like within that year prior to your soil sampling event, because we're getting some strange or some numbers that make sense if they've been fertilized, but don't make sense if they haven't. And then we start to wonder if something's wrong with our analytical equipment. So we just kind of need to know that for context. Yeah, and one other thing, while these soil test results are really awesome and great to track because you've got the numbers, it might take a while for you all to get these back because it takes almost a month to get through all these analysis. But we do have the Rocky Mountain Region Infield Soil Health Guide, which is something you can go out and do with your producers, and you can actually track like visually and other like tests you can do out on the landscape with them, how their soil health is changing or looking over time. And in some ways, I almost feel like an infield assessment is more informative because soil health a lot of the time is something you can see and not always something that a number totally reflects. So the numbers are very important. So the QR code should take you to this. It's on the Enrich's website. We can send you all out other links to click through. There's something like, I think, 17 different infield assessments that walks you through the steps and all the equipment, and there's different versions of it. Is there a question, Jen?
Yeah, so I had done the sampling for my area, so I have all my surveys inputted already. Is there a certain way that you want to collect that information for that additional question on fertilization? Like a certain email I should just check in with my producers and then send it to you?
Yeah, we just decided this morning, so I'm gonna
okay.
I'll have John
You'll get back to us.
mail you out like what we're gonna do with that, because we know that there's been over 100 soil samples sent in, and we were like, so yeah, we'll get back to you on that. I just wanted to let you know future ones there is going to be another one on there.
Thank you.
Just became an issue.
That would be great, Jed. Just a really quick point on that, just so you all understand. So there's one aspect of like the quality assurance, quality control while we're running and analyzing these samples for the ammonium and nitrate that Wilma talked about. Another thing will be in the interpretation of those values, both for when you take them back to the producers and understand the levels of ammonium or nitrate and whether that was included. Likely it was included if they had fertilized their yards or their fields. But another thing too is when we're going to be, we at CSU are synthesizing all of this data to really help us advance the science of soil health. And Wilma mentioned this maybe unique to most soil testing reports that you get back. We're analyzing the potentially mineralizable nitrogen. And you can kind of think of that as your fertilizer savings, potentially. In the Haney report, I think that often producers use, you get something called a fertilizer savings. Now that is not analyzing mineralizable nitrogen. That is estimated off of soil respiration. So we really have the unique opportunity here to really understand how these soil health practices that are being deployed are potentially improving that bank account of nitrogen that is microbially mineralized, therefore could be reducing the amount of fertilizer that a producer may need. So that context, understanding if they fertilize or not, will be important in various ways. And I just wanted to make sure to expand on that a little bit more so that you know kind of why that piece of information is needed. And of course, as we're going through this, you know, we're learning as we go, and as we're looking at these values and thinking, oh, well, probably some producers have fertilized and that's why it's coming up now. So that's just a little bit of context of why that would be good to have. And we'll figure out the most efficient way to get that information from you. But I really, truly appreciate, Jen, you being able to, you know, send that in, even though you've completed all the surveys. Just want to give a little context there. Thank you.
And I wanted to jump in here real quick on these in-field soil health assessments. Our CDA soil health specialists do have some in-field soil health assessment kits. It's, you know, it'd obviously be really hard to do these for every single producer, but we'd be glad to run some of these assessments for you. Might be good things to demonstrate at field days and things like that. So we do have those kits. And we also have a list of supplies for conservation districts and entities that want to build their own. So if you're interested in that, please reach out to us.
Thanks for mentioning that, John. I'll just note, for those of you on the Front Range, Larimer County is holding a field day where we will be handing out hard copies of this in-field soil health assessment guides. And I think Lexi, or I can actually, since you have the presentation up, I can put the link, if I can find it quickly, to that field day so that those that are interested, if you haven't registered already, you can register for that. And I can't remember the date I think it's this weekend, this Saturday, correct, Lexi? Or is it 2 Saturdays from now?
It's 2 Saturdays from now, the 20th or 22nd, or it's the winter's, or it's the weekend of the summer solstice. So that's Saturday.
Great.
Whatever that is.
Great. Okay, I'll try to find the link and put it in there for those in the chat for those that are interested.
Mm-hmm.
But of course, this is always available online on our website and via this QR code. And at the end, we can share this presentation with you all. But at the end, we have a slide with links to various resources. So you can use that link as well.
And using the link is actually really helpful because there are like actual linked websites and YouTube videos if you have it online. So like having a hard copy is great with the field, but if you actually want to go directly to a website or something, it's nice to just know where that is. Okay.
Hey, Lexi, we have a question real quick before we move on. Now, just remember, anything that anybody does as part of the CDA soil health program needs to be tracked. So if they're applying anything on their fields, we need them to keep track of that, and that'll be part of their reimbursement package. But specifically, I think, Lexi, to answer your question, for the future soil samples, what fertilizer do you want to track? You'll probably have a really nice, easy question in the survey 123 that you click. Is it organic? Is it non? Is it manure, etc.? And so that should be pretty self-explanatory, I'm guessing.
We are also working on an easy nutrient management document to help to gather the metrics of what people are applying and when they're applying it. We'll be getting that out to you. As far as like, we do want you to track, you know, organic fertilizer, Penny, but it's hard to track the actual amount of manure that's maybe left on a field after you have cattle grazing out there. But if they're like applying manure with a manure spreader, that'd be a different situation where we'd want to know, you know, how much they're applying and things like that. And we are, so we're working on a way to track this. So.
And that was my question, because we do have a producer that does graze during the fall winter season. So how to track that is probably very hard to do.
Yeah, I don't think that's something we probably need to try to track, but yeah, I'd be thinking more of something they're applying, you know, with a spreader or broadcaster or something like that.
Thank you.
Yeah, so the question on the form will probably be a very simple, did they apply fertilizer? Yes or no. And then CDA will have their very detailed what you applied, when you applied it, information that we can collect. But as it's just easier as we get it in, quick check. Yes, they did. Okay, then this value we need to just know. for context. It's okay or it's not okay. We're getting really short on time, so we are measuring a few other things. I don't know, Megan, if you want to talk about this, but I do want to give the microbiome folks some time.
Yep. No, I think it's fine to move on. I think you're all familiar with, and you're helping install these soil moisture monitoring sensors. Those of you that were involved last year your producers received an annual report that had color-coded graphs like these that just quickly demonstrate, the green zone is a great sufficient zone for plant water uptake. And then we go through that report and define field capacity and saturation and wilting point, which is the red. But just a note to remind your producers that they do have this at their fingertips on the Zentra Cloud app on their phones. But let's go into microbiome details now.
If they do not know how to access their Zentra Cloud app or are not able to get in there yet, please let us know and we'll get you a link. This can't be understated. All right.
Yes.
I'll shut up now.
Yeah, me too.
Laura, would it be easier if you shared your screen? I know we have the same PowerPoint, or do you just want me to click through for you?
I think Chris is gonna lead the way on this one, so kind of whatever he wants. Maybe just clicking through because we're short on time though.
I've got a couple animations, so I'll just tell you when to click.
Okay.
But yeah, thanks for having us. Again, I'm Chris Corpenning and here with Laura Mason. So we're both part of the Microbiome Ecosystems Lab, and that's spearheaded by Kelly Wright and Caleb Borton in the Soil and Crop Science Department. So our goal is really, in general, to understand kind of how microbes and the microbiome impact and define ecosystem functions. So things like how greenhouse gases are, emitted from soil, but also sequestered and kept in there, and how those things changes. That's all kind of down to the microbiome. Things like how nutrient cycles, like how nitrogen is transformed and what happens to it, sort of the fate of that nitrogen in the soil, as well as the microbiome of our own guts. That's of huge interest too. And now we're really curious about how the microbiome really matters for soil health and all the different aspects of it. We've talked a lot about the biological aspects of soil health today. And so now we're at a point, scientifically and technology-wise, where we can really more directly measure kind of what's going on with the biology of the soil. So yeah, there we go. And so kind of just wanted to briefly talk about what a microbiome really is. It's kind of a buzzword now. So the cold definition is it's really a collection of microorganisms, so like bacteria, all of their genes, so their genetic information, like in their DNA, but then also considering the chemistry of their environment. So the different chemicals there and how those affect those microbes, but also how those microbes affect the chemistry, sort of that back and forth there. And so we look at their DNA, we look at what their genes are telling us. And so through that, we can kind of see who's there. We can see the identity of these different fungi, these different bacteria. We can also look into what they're doing, so what they're eating and what these different nutrients and chemicals are that they're processing. And we care about those different processes that might matter for, plant productivity, for emission of, methane or other things from soil or other environments. So yeah, go to the next one. So very importantly is the why. Why do we do it? We really are concerned about discovering sort of the underlying engine for how these different nutrients like nitrogen, carbon as well, all these things are moving through the environment to the soil. Really what's making soil alive. A lot of these aggregates which we measure, we care a lot about, are really influenced heavily by microbes, the fungi and bacteria in the soil. And so we also kind of understanding or trying to gain understanding on how different ways we can measure the microbiome might be able to inform soil health testing in the future at local, but then also regional levels. We know that obviously our practices here in the West differ from the Midwest and the East Coast and such. And so understanding how the microbiome is inherently different and how they're different in our management systems is going to be really important for understanding what they might mean for soil health for us. And so we are really trying to answer questions about how the microbiome matters to soil health. It's still a pretty cutting edge area of research in general within the microbiome, and we're more recently able to apply it to agriculture and soil health. And so we're kind of asking things like what groups of microbes matter the most, like those specific, are there specific ones, groups of microbes that, seem to predict soil health. And yeah, finding those links between what we measure in the microbiome and then the different factors of soil health. Like we've talked about potentially mineralizable nitrogen. Those are microbes that are processing that nitrogen. Are we seeing kind of a link between the microbiome that we're measuring and then kind of what we're seeing from other tests from these soils? And then ultimately, I think we're probably a ways out from this, but who knows? Are we able to prescribe management and really measure soil health and predict future soil health based on the microbiome and using the microbiome as sort of a prescriptive tool? That's sort of the stretch goal, the ultimate thing. I just wanted to talk briefly about some of the current microbiome metrics and measures that are offered in some soil health testing commercially, I think. So these may be things that some folks or producers have seen. Maybe you've been asked to interpret or look into them. Oftentimes we'll see things like the fungal to bacterial ratio or like fungal and bacterial diversity kind of along with that. It's generally thought that higher like fungi to bacteria is better. It's a relatively broad assumption and is, kind of questionably credible in some cases. And it can also be really, system and specific, or system and regionally specific. What's good for a pine forest and a grassland is not necessarily what's good for, an irrigated pasture or irrigated corn, right? And it's a bit more complex than just a ratio between these two very different groups, which are very diverse in themselves. You might also see some like enzyme assays, and so that kind of aims to look more at what is actually being done by the microbiome, some of the processes that they're carrying out that might be digesting carbon or nitrogen in different ways. And so this can provide some of an overview of like some of these processes. But they're definitely, when you're only testing 5 different enzymes out of, thousands which are being produced, you could be missing some pretty vital, important things. And so that's why we're really hoping, and now technology is allowing us to do more genetic investigations. So we're able to look at, DNA to understand more of the complexity and the specific identities of bacteria and fungi and other microbes that can help provide us better insights into how the microbiome is made up and what it can really do and how that impacts soil health. And I think instead of just looking at, a ratio, we're looking at a few specific enzymes in the soil. We try to take a more holistic view and look at, the whole community. I love this line, it's a microbial community effort, and it really is. We are, ecologists at heart, so we care, and we're really concerned about this entire function. I think that's what soil health represents too. It's looking at big picture. And so, yeah. It's a little bit about kind of our collaboration with CDA and with Lexi and Megan. We're just really excited that we get to, further this cutting edge research. There we go. Yeah, trying to understand just about specifically here in the West, what microbial content, the microbes in the soil, what it tells us about soil health. Are there things about the microbiome that predict soil health that are positive for it, or are there things that sort of contradict it? Okay, we observe the microbiome this way, and those seem to be frequently in poor soil health situations. And then if there's kind of a through line we can identify in our region, that can be, a good test in the future potentially. But we have to look first, so that's what we're doing. And so just a bit of what we'll be able to see from your samples without getting in the weeds too much of different kinds of microbiome testing. Broadly, we're looking at who's there. So we're looking at the identity and the different types of fungi and bacteria in the soil. And from there, we can kind of infer as to what their function is. What we kind of have, there are some bacteria and fungi which have a very, conserved range of abilities and of functions they can do in the soil. So there's some that, really, we know they process nitrogen in this way. And so they are indicative of, nitrogen loss sometimes. We also are looking at microbiome structuring. So a little more kind of ecological take there and sort of how the microbiome is assembled and in what different proportions these microbes are together. And then we can also kind of look at who's connected to who, who microbes are associated with others. And sometimes those different dynamics can show us a little bit more about soil health than even just the identity of them. But we're definitely still, learning what the microbiome means for soil health. It's a really complex area, as is soil health in general. And so we're still, kind of developing these more meaningful outputs and learning what these things mean. And a lot of this, we're really working towards building out something bigger. And that's why this project is really exciting. So I'll let Laura talk about on this slide.
Yeah, so thanks so much, Chris. Yeah, like Chris was saying, we're working on building up our microbiome knowledge of the West and beyond. So on the left here, we have a diagram of what some of our field studies would look like from a microbiome and computational perspective. So we can do field studies and compare the results of these field studies to existing and future databases that we're working on. And then we're also working on building these databases and these computational tools to cover microbiomes throughout the Mountain West. The panel on the right shows all of your sampling points. The red ones are CDA star producer points. These purple ones are the Enriches team's points, and these kind of dark greenish blue ones are our lab's points where we already have microbiome data in hand, and all this is going together to build functional and taxonomic profiles of the microbial community in this region. We really just to set kind of benchmarks for baseline or I guess like baseline microbial activity and how that relates to soil health specific to this region. So yeah, that is what we're doing, and we're happy to talk more about it at a later date.
Sorry.
Awesome.
Yeah, and sorry, we're going over last slide. Just context, we've got this microbiome database that potentially is being built. We've got all these soil moisture sensors that are going out to build this soil moisture monitoring network. And then all the soil health data is also coming in to build this other very large database, which we're hoping that we can somehow merge into a soil health discovery platform that can give region-specific soil health decision support tools to producers. And I could talk about this forever, but I don't want to keep you all too long. So I can hang on and answer questions, but other than that, that's what we got for you today. All of our contact information is here, and we can send out these slides as well, because there's also some helpful links that I have here. And yeah, that's it. Megan, unless you wanted to add something.
No, thanks all for your attention. Sorry we went over a bit. I'm sure we could have multiple sessions here to dive into the details, and we can certainly, if there is requests, we can think about, going into details on any of this in the future. But yeah, I'm happy to hang on a bit for questions if there are any. And thanks for having us today.
Thank you all. This, I wanted to just underscore, this is a really unique opportunity we have here. We have all of this data we're gathering, these really in-depth tests, the microbiome data, and then we also have all this information about these different practices that are occurring. And we have the opportunity here to really cross-reference these things. And I think there's a lot that can come out of this. And, we wouldn't be here having all this happen without all the hard work of the people on this call. So, and thank you to the folks who have presented and are making this science happen behind the scenes. So thank you guys. It's pretty quiet. I think this is